right, so it is a great pleasure to introduce Adrian Isaacs, who's going to give the second talk. So Adrian is a group leader at the UK Dementia Research Institute, and he's also the Professor of Neurodegenerative Disease at the University College London. Adrian has a long-standing interest in ALS and FTD mechanisms. He's specifically focused on two distinct genetic subtypes of FTD and ALS. He is really interested in CHM2B as a cause of FTD, and he's also made some seminal contributions in our understanding of how C9orf72 repeat expansions contribute to neurodegeneration in the context of ALS. His group was one of the first ones to show that the repeat expansion gives rise to antisense foci. He was also one of the first groups to dissect the contribution between expanded RNA and dipeptide repeat proteins. And I'm delighted that he's here today. I think he's going to talk to us about C9orf72 today. The title of his talk is New Disease Mechanisms and Therapeutic Approaches for C9orf72 in ALS. So Adrian, welcome, and thank you.
Thank you. I just wanted to start by thanking the organizers for inviting me to this super interesting symposium. And I've already been learning about the wonderful work of the Les Turner Foundation to support ALS research here at Northwestern, but also the sort of wider ALS community in Chicago. So really it's a pleasure to be here. So as Evangelos said, my lab has really focused for many years now on trying to understand genetic causes of ALS, and one of our major focuses has been C9orf72. So what I'd like to do is give you a little bit of background on C9orf72. I'm not really going to go into ALS itself because I know you're all experts on ALS, and just tell you about a bit of our early work that's really led to these two very recent publications that both came out this year that I want to go through. One about a new gene therapy approach that we're working on, and another really uncovering new neuroprotective roles for lipids in ALS. So, as I'm sure many of you know, a repeat expansion in C9orf72, so an expansion of 4 Gs and 2 Cs, is the most common genetic cause of both FTD, frontotemporal dementia, and ALS. And it actually accounts for about 10% of all cases of FTD and about 10% of all cases of ALS, so this is clearly an important and frequent cause of neurodegeneration. And the repeat expansion occurs in the first intron of C9orf72, and we have an arbitrary cutoff of 30 repeats, but actually most people, the majority of people, have just 2 repeats, so 4G2C, and then another 4G2C, and then carrying on with the gene, whereas in patients, if you look in the brain, then the repeats are generally several hundred and usually in the thousands. So you have this very large extra trunk of DNA. And this has led to many different mechanisms being put forward. So the first one I've listed here is RNA toxicity, and this is really taken from the myotonic dystrophy literature. So in myotonic dystrophy, which is another repeat expansion, we know that the RNA that's transcribed from that repeat aggregates in the cell and can sequester RNA binding proteins, and it's the repeat RNA itself that is definitely contributing to neurodegeneration. We also know, through really pioneering work from Len Petroselli and Dieter Edbauer, but building on the truly pioneering work from Laura Ranum, that these repeats can escape into the cytoplasm and then become translated into proteins. And because you have a repetitive DNA, it makes repetitive RNA, it makes repetitive proteins, and we call them dipeptide repeat proteins. So in addition to that, the repeat expansion can also reduce the expression level of the normal C9orf72 protein, and it's likely or possible that reduction of the normal C9orf72 protein may contribute to the disease process. So we used to say that was it. There were 3 disease processes, but now this is a new slide. I've updated it. We've now got 4. So actually, the more we learn, the more complicated the picture becomes. And that's because there is some emerging evidence suggesting that perhaps at the DNA level, the DNA repeat itself can also cause toxicity through things like epigenetic changes, or even potentially by initiating innate immune responses. So there's clearly a plethora of different potential mechanisms. My lab has really been focused on those first two, the RNA toxicity and the protein toxicity. So in some of our early work, we were really interested in that myotonic dystrophy literature and trying to understand whether the repeat itself, whether the repeat RNA itself, I should say, was aggregating. So what I'm showing you here is a zoom of a single neuron in the brain of an FTD patient with a C9orf72 mutation, and we've made a fluorescent probe against this 4G2C sequence, and you can see these aggregates, or foci as we call them, of RNA, these red aggregates, and you never see those in control. So this is very specific for the C9 repeat expansion, as you would expect. But I think the important part of this paper was again inspired by the myotonic dystrophy literature. We decided to see whether the repeat was also transcribed in the antisense direction. So we made probes against this antisense sequence. So instead of 4 Gs and 2 Cs, this is 4 Cs and 2 Gs. And indeed, we saw really numerous aggregates of the antisense repeats. And the reason I think this was an important paper at the time is it suggested there may be this additional pathogenic entity that we now need to consider, not only the sense-derived pathologies, but also these antisense-derived pathologies. And as I'll go on to discuss, I think that is really coming back into focus now as an important area of research. Okay, so while we were doing that, this parallel work was going on investigating not the repeat RNA, but the repeat proteins. And as I've mentioned already, several teams showed that these repeats can undergo this remarkable phenomenon called repeat-associated non-ATG translation, or RAN translation. So as you know, normally proteins need an ATG start codon to initiate, trial ribosomes need an ATG start codon to initiate translation of an RNA. But these repeats can mediate their own translation into proteins. So what that means is the repeats can be generated, or the repeats can be translated in every frame. So just to walk you through what this looks like, if you have 3 Gs here, that will make a glycine, and then GCC will make alanine, and you get a repetitive glycine-alanine sequence, so this dipeptide repeat. If you move one base pair along, in the next frame you still get 3 Gs, which makes glycine, but now where are we? Is it GCC or CCG? That will make proline. So you end up with polyglycine proline. And if you move one base pair along again in the final frame, you make polyglycine arginine. You also get these proteins derived from the antisense strand. So now we end up with multiple potential pathogenic species. And it's been clear, and it's from the data I'll show you, that poly-GR on the sense frame clearly exerts some toxicity. Poly-PR, which comes from the antisense frame, also exerts toxicity. And I'll also talk a little bit about poly-GP, which seems to be more benign, but is the easiest to measure. So it's been very useful as a biomarker that you can measure in patient biofluids. Okay. So when you make antibodies against each of these dipeptide repeat proteins, and then look in patient brain, again here we have a zoom of a single neuron. In each case, you can see these aggregates form that are derived from these dipeptide repeat proteins. So we know that these aggregate in the patient brain. So a lot of my early work, as Evangelos said, was trying to tease out these different pathologies and see which may be contributing to disease. So I want to walk you through just a couple of examples of that early work, really to introduce the models that we use in my lab. So in some of our early work, we really focused on using the fruit fly. And the fruit fly has been really an incredible engine of discovery for us and initiated a lot of the work that we then take through into our human systems. So here, what we've done is take repeats, so 100 GR or PR repeats, and express them in the fly eye. So this is a zoom in of the fly eye here. So this is a normal looking eye, and you can see that 100 repeats of either GR or PR is really acutely neurotoxic. The second paradigm we use is we take a neuron-specific inducible driver to turn on repeats only in adult neurons in the flies. And then we will do a survival experiment. So in the survival experiment, in the survival experiment, we'll put about 150 flies or so in this, and then we will just see how long they survive. So flies in the lab can live up to 100 days. It's quite surprising, and you've got your flies buzzing around your fruit bowl at home. Actually, in the lab, we can keep them for 3 months. Whereas if you express GR or PR, you can see they die extremely rapidly. So clearly, GR and PR are sufficient to cause neurodegeneration. And again, the point is, GR is encoded by the sense frame or the sense transcripts, and PR is encoded by the antisense transcripts. So this was data that we produced very early as my lab was just starting, and a long-term goal of the lab had been to try and see whether we could replicate these findings in a mammalian model, but also not using the kind of gross overexpression that we're using here. So to do that, we generated knock-in mice. So here we built 400 repeats of either GR or PR, and then using CRISPR-Cas9, we inserted them immediately downstream of the mouse C9orf72 ATG start codon, so they could be driven by the mouse C9orf72 promoter, so we could express them at the right time and in the right place, and ideally at more physiological expression levels. And I'm not going to take you through everything that we did in this paper. We published it last year, but this is just to show that when we measured motor neurons in the spinal cord of these mice, even though at 6 months there's no difference, by 12 months there was about a 20% reduction in motor neurons through expression of either GR or PR. And just to confirm this, we then did some electrophysiology with some collaborators at UCL, and using electrophysiology as well, using this motor unit number estimation, we can see again that there's a reduction in functional motor units in our GR and PR mice. So this really is suggesting that GR and PR are sufficient to drive neurodegeneration, not only in the fly model, but also in a mammalian system. So that leads on to this point, which is, do we need to be targeting both sense and antisense repeats to have an effective therapy for C9? And there's a very active discussion going on. I'm sure that many of you are aware that, it was a great disappointment for the field when these two antisense oligonucleotide trials, one led by Ionis and Biogen and one by Wave Life Sciences, both failed in clinical trial, even though there was target engagement in that, at least in patient biofluids, you could see a reduction in these sense-derived dipeptide repeat proteins. And I think there are actually many potential reasons for that, and we discuss a few of them in the reference I give at the bottom, but I think one potential explanation is that these antisense-derived repeats weren't being targeted. And for me, I think this is a high priority for the field now, to interrogate that possibility that the antisense repeats are driving some toxicity. And I think it's important that we start moving therapeutic approaches that are going to target antisense repeats into the clinic. So as we had discovered the presence of those repeats a while ago, we'd been thinking about ways that we might try and do that. And one approach that we're working on is to use CRISPR-Cas13. So many of you will have heard of Cas9. So that's DNase that cleaves DNA. So Cas13 is similar, but instead of a DNase domain, it has an RNase domain, so it can cleave RNA. But the reason I got really excited about Cas13 when I was reading these initial papers about it was that it can cleave its own guide array. So what that means is the way that Cas13 works is a guide RNA will take it to the transcript that will then degrade, but as it can mature its own guide array, you can load it with more than one guide, so you can target more than one transcript at the same time. So it felt like potentially a way that we could target both sense and antisense repeats. And this is a project that was really led in the lab by Liam Kempthorne and Denise Weissoglu on the in vitro part, and then Alex Kamek led the in vivo part. So this is a kind of short potted history of Cas13. So they were identified in the 2017s and 2018, but there was a particular interest in Cas13D, also termed CasRx, so I'll call it CasRx. And that's because you can see from the graph that Cas13D is much smaller than the other Cas13s, and indeed the original Cas9s as well. So it's small enough that you could fit it into a single AAV. So we could imagine a potential therapeutic where you could fit Cas13 plus guides to antisense and sense into a single AAV. So you could have a one-shot treatment potentially for C9. So this is something we wanted to investigate. So we started with an in vitro assay in hex cells where we built a reporter. So we put some 92 C9 repeats, and after it a nanoluciferase, but the nanoluciferase doesn't have a start codon. So you only see nanoluciferase expression if you get transcription through the repeats. So it's a nice way of measuring the repeat levels. So we tested a couple of different Cases, and Cas13b you can see had a modest effect, whereas CasRx was really extremely effective in knocking down this repeat transcripts really to a background level. And then we did a final experiment where we took dead CasRx. So this is a bit like dead Cas9. It doesn't have enzymatic activity. And we thought that might be sufficient, actually. We thought if you could get the Cas protein just to sit on the RNA, that might be enough to just prevent translation. But that turned out not to be the case. So you definitely need the enzymatic activity of CasRx to drive this reduction in dipeptide repeat proteins. So this was in the sense direction. So then we did the same thing in the antisense direction. We built an antisense reporter. And again, we found several guides that were really very effective at reducing the antisense transcripts in this reporter. So then we wanted to put this together because the idea was that we would put it in a single guide array. So these experiments are testing whether indeed you can add CasRx with a guide array with guides targeting both sense and antisense. And as you can see, that also works. So here in our sense assay, if we have guide 10 on its own, it's very good at reducing sense repeat levels. But in a dual guide where you have 10 and 17 together, 17 is our antisense guide, you still get very effective knockdown. And the same is true in our antisense assay. Guide 17 knocks down very well, but in a dual guide approach, you still get very good knockdown. So then we went on to look at this in patient iPSCs. So here we use this system that many people are using where you add an NGN2 cassette, which means you can then add doxycycline and very rapidly induce neuronal differentiation. And I know many of you use these all the time. I just love this video, so I just wanted to put it in, seeing this really beautiful rapid differentiation of neurons. So using this approach, we then put CasRx into our patient iPS lines, and again we saw pretty good knockdown of all of the different species we're interested in. So again, we can see knockdown of antisense transcripts, and it is a bit variable between different patients, which I think it's really important that we use multiple patient lines when we're doing this type of work. You can also see a reduction in sense transcripts and also a reduction at the protein level in poly-GP here very consistently. So that gave us confidence to go into mice, and we used two different mouse models. So the first one is an AAV mouse model where we use a first AAV that has 149 repeats. It was kindly provided by Len Petrocelli to induce disease. And then we co-inject with a second AAV that has our CasRx and our dual targeting guide construct. And what you can see here again in the graph on the right is that indeed we see a reduction in sense repeat containing transcripts. So this particular mouse model doesn't have much flanking sequence for the antisense transcripts, so we couldn't target the antisense transcripts in this mouse model. So then we moved on to a second mouse which is the C9orf72 back transgenic mouse, which has now the whole human transgene and about 500 repeats. So here we can target sense and antisense together. So this is a slightly simpler experimental paradigm where we just inject the potentially therapeutic construct, and then we waited nearly 9 months in this case, and then looked for reduction in expression. And you can see again that we see a reduction in sense repeats, we see a reduction in antisense transcripts, and a reduction in GP. The reduction is more modest, but that actually reflects our transduction efficiency, so that we know that in the cells in which we have delivered the Cas, we get very good transduction. And it's just a reminder that delivery is really the key to taking these forward, I think, and to making them effective in the clinic. But just as a little side story, we'd become very good at measuring these dipeptide repeat proteins like poly-GP in our mouse and iPS samples, and we really wanted to see whether we could take that forward into patients. And that's because if we're really truly going to think about translating this to the clinic, we need to have a sort of biomarker plan in place. So this is some work we did initially with Henrik Zetterberg, who's really a leader in biomarker development. So we were co-located with Henrik at the time. So he had several Simoa machines in his lab that we could have access to. So the Simoa platform is really a highly sensitive platform for detecting proteins and is widely used now for NFL. And so we basically took our lab-based poly-GP assay that was an MSD immunoassay, and we switched it to the Simoa platform, and then working with John Rohrer, who leads the genetic FTD initiative, we measured poly-GP in the CSF of really a large number of C9 FTD patients. And what you can see is both in pre-symptomatic and symptomatic patients, we can see poly-GP, and we never see it in controls. So in this particular cohort, we had 100% sensitivity and 100% specificity. So this is really an excellent assay. And because we have the sensitivity of the Simoa, you can see even the patients who have very low levels of poly-GP, there's a sufficient window there that would be able to see target engagement and see a reduction. So we then went and worked with Wave Life Sciences to do some additional assay validation to get this ready for clinical trial samples, and then we used it in the Wave Life Science ASO clinical trial. So it was a really nice example where we took our lab assays all the way through to patients. And of course, you know, it was disappointing that there was no effect in that trial, but we know we have a good target engagement biomarker now moving forward. So just to summarize that part of the talk, we've shown that CasRx is effective in vitro and in vivo. We have a sense repeat target engagement biomarker in place, and I think something that we're actively working on, I think something that's going to be really important for pushing the field forward, is developing now target engagement biomarkers for those antisense repeats. Okay, so now I want to change directions and talk about our lipid story. So this, as I said, started with the fly and was really led by two really brilliant lab members, Ashlyn Giblin, who was a PhD student who's now graduated, and Alex Kamak, who's a senior postdoc in the lab. So this started with a fly experiment. Here we're expressing the repeat expansion in the fly, so 4 Gs and 2 Cs, 36 of them, and again you get this rapid death. So we decided to do an RNA-seq experiment at an early time point to try and understand what some of the early changes were. And we were struck when we did this gene ontology enrichment analysis that many lipid terms were coming up. So when we drilled down in this in a little bit more detail, it was really clear that nearly every or many genes in this long-chain fatty acid synthesis and desaturation pathway were downregulated. So I just want to walk you through this because I'll be talking about it a bit now for the next part of the talk. But this is long-chain fatty acid synthesis. Acetyl-CoA ultimately ends up in palmitic acid. So we call that 16-0 because it'll have 16 carbons and no double bonds. Fatty acid elongation will add 2 more carbons and you'll get stearic acid, which is 18-0, so 18 carbons and no double bonds. And then a desaturase enzyme can add a double bond. So the addition of a double bond is an unsaturation event. And then, in this case, this fatty acid would be monounsaturated, and if you'd add more double bonds, they'll be polyunsaturated. But just to give you an idea of the importance of these saturation events of adding double bonds, the stearic acid is solid at room temperature, whereas oleic acid is the main constituent of olive oil. That, of course, is liquid at room temperature. And actually, the addition of that one double bond changes the melting temperature of the fatty acid by over 50 degrees. So really, it has a very profound effect on the physicochemical properties of the fatty acids, and saturation is something we've become very interested in now. So this was like a transcriptional suggestion there may be something going wrong with lipids, but then we wanted to go on and, of course, see whether that was the case. So we're very fortunate to work with Martin Gera through the Chan Zuckerberg initiative. We linked up with him, and he's been a wonderful collaborator to do lipidomics with us. So we sent him the fly brains of our C9 repeat flies, and he did the lipidomics. So he does a targeted lipidomics. It's 400 different lipid species. And we didn't see global changes, but where we saw some effects was in phospholipids. So I just want to again walk you through and remind you what a phospholipid is. So it has a hydrophobic head group, and then it has these hydrophilic head group and hydrophobic tails. And those hydrophobic tails are, in fact, those long-chain fatty acids that I've just been describing to you. So we've got here an example of a saturated fatty acid, and in the second tail, 2 double bonds here, so that's a polyunsaturated fatty acid. So when we measure this one way, we look at this as doing a heat map. So here in the heat map, we've just basically plotted all of the different phospholipids, but we've ranked them by the number of double bonds there are in those fatty acyl chains. And what you can see is that as you go down here, it gets much bluer as you have more double bonds, so more unsaturation, suggesting a reduction in these highly unsaturated fatty acids within phospholipids. And that's probably easiest to see here in the volcano plot, where if we color the phospholipids by either having 0 or 1 double bond or 2 or more double bonds, you can see that when you have 2 or more double bonds, they're really reduced in the C9 fly brains. So then we wanted to go on and check this out in patient I3 neurons, so using the same system that I've already described to you. But because I3 neuron or patient neurons can be quite variable, we wanted to use 3 different paradigms to really understand what was driving any effects we were going to see. So the first was to use the sort of standard paradigm where we take a control, we take C9 patients and we compare them to isogenic controls. And one way we sort of verify our lines is to measure these dipeptide repeat proteins, and you can see all of our lines make a good amount of this poly-GA. So the second thing we then did is take a slightly different approach where we take a control line and overexpress using a lentivirus. So LV here is a lentivirus, either 2 repeats as a control or 92 repeats. So this is really asking, are those repeats sufficient to drive any effect we see? And again, you can see when we add 92 repeats, we now get good expression of dipeptide repeats. And then finally, we did kind of the reverse, where we took an antisense oligonucleotide and we fed it for 18 days to our patient cells, and it targets the repeat expressing transcripts. And you can see using an 18-day treatment of the ASO, we really get very dramatic reduction of these dipeptides. So we're really, again, asking, it's a specificity question, are the results specific to the repeat transcripts? And what we saw was that when we do this same lipidomics and we do this heat map again, in the patient lines versus the isogenic controls, you see a reduction in these highly unsaturated phospholipids. If we overexpress the repeat, we see exactly the same thing, but slightly stronger, as you might expect, saying that the repeat is indeed driving this effect. And then if we treat with an ASO, we see the opposite effect, showing again that it looks like the repeats are driving this loss of highly unsaturated phospholipids. So then we looked in patient material, working with Tamarin Lashley, who's a long-time collaborator at UCL, and we looked in a large number of FTD cases, both in the frontal cortex and the cerebellum. And again, while the cerebellum was less clear in the frontal cortex, which is really the area where we see degeneration, we saw this similar pattern of this reduction in highly unsaturated phospholipids. And I always think it's slightly difficult to interpret the post-mortem data, which is really the end stage, but it's telling us that there's something going on with lipid saturation in the disease. So then, having all this descriptive data, really the power of the flies is we can then go back and start asking some mechanistic questions about what is the consequence of this. Is this beneficial? Is it harmful? Is it neutral? So just to tell you about this set of experiments, I've just got to walk you through a little bit more of the desaturation pathway. So I think we got to oleic acid 18.1, but if you add another double bond, you get linoleic acid 18.2, or here 18.3 with a third double bond. And these are actually quite well known, and they're talked about a bit in the kind of supplement industry because they're known as essential fatty acids. And what that means is the body can't make them, you have to get them from the diet. So the most simple first thing to do was to take these and feed them to the flies. And so here's our survival assay. And what you can see is both 18.2 and 18.3 both shift that survival curve a little bit, so they're beneficial. So that's suggesting that they can, it's suggesting that this loss is harmful and that giving some back is beneficial. But of course, these are quite modest effects. So we wondered whether there was a way, rather than through the diet, we could get more into the brain. So this is the experiment I love most in this study. I really love this experiment. So we use the kind of quirk of evolution that even though 18.2 and 18.3 are essential for us and they're essential for flies, they're not essential for worms. So worms have an enzyme, a desaturase enzyme called FAT2, that can actually make these two different lipid species. So then we could take that enzyme, we could give it again a neuronal, run it under a neuronal driver and turn it on only in the neurons of the flies, thereby delivering 18.2 and 18.3 only to the neurons in flies. And the power of having lipidomics is then we can see this is absolutely happening when we do this. So we overexpress FAT1 and FAT2. FAT2 in this case, and you can see that the substrate 18.1 is reduced and 18.2 and 18.3 are increased. And then when we do this, we see this much bigger increase in survival, suggesting that these fatty acids are beneficial, but it's really getting them to the brain that's providing that benefit. So again, then we wanted to do a similar experiment in our iPS neurons. So here we're overexpressing FAT2 in iPS neurons. And again, the lipidomics shows you that when we do that, we get an increase in 18.2, an increase in 18.3. But in this case, in the neuronal cultures, that really feeds forward into these species that have even more polyunsaturated, even more unsaturation, so 4, 5, 6 double bonds. And then to understand the consequence of this, we worked with Alyssa Coyne at Johns Hopkins, who has this very nice assay where she takes 6 C9 patient iPS neurons, compares them to 6 different controls, and takes them out to about 32 days of differentiation, and we're calling them SNs, so spinal motor neurons, and then she challenges them with glutamate. And what you can see when she does that, if you look at the control lines here, the C9 lines are more sensitive to glutamate challenge. So this is essentially a cell death, amount of cell death. But when we add FAT2, there's a reduction in cell death, suggesting that in the patient iPS neurons, increasing this unsaturation is beneficial. So that's all the data I want to show you. But the reason we think this is important is that there is clearly epidemiological evidence suggesting that these polyunsaturated fatty acids can reduce the risk of ALS. So these are both studies from Alberto Echerier's group at the Harvard School of Public Health. So one published over 10 years ago was a prospective study. It followed a dietary questionnaire to try and understand how much polyunsaturated fatty acids were being taken up. And of these million people that were followed, about 1,000 people got ALS. But a lower proportion of people got ALS in the group that had the highest intake of polyunsaturated fatty acids. So that's dietary questionnaire. And then in more recent work, they did a different study where they measured the amount of polyunsaturated fatty acids in the blood of people with ALS. and then followed them over time. This was kind of an add-on to a clinical trial. And what you can see here is measurements of ALA, so 18.3. And in the quartile who had the highest amounts of ALA in their blood at baseline, this is incidence of death. There's a reduced risk of death. So based on this data, the Asheri group are really convinced that there's enough evidence to try a clinical trial now to use 18.3 in this case to see whether that would provide benefit for patients. And based on our data, we're very supportive of that. And there's ongoing discussions about whether that may happen through the Experts ALS platform in the UK. And I think the point to take here is I think this is definitely low-hanging fruit that should be investigated, that simple dietary intervention may provide some benefit. But you can see from these graphs, of course, that ALS is relentless, and whatever you do, patients are still dying. So I think, while I think this is really important to do, of course, this simple dietary intervention is not going to be enough to completely stop ALS. But I think what our data is telling us is that actually getting these into the brain is going to be the most benefit. So while the dietary intervention may be the first thing that we should try, if we can really understand the mechanisms of benefit in the brain, we might be able to develop better drugs that can actually mimic that process more effectively. And that's where we want to take the research next. So I'll just finish there with this summary. I've shown you it's this loss of highly unsaturated phospholipids. We think promoting their levels ultimately is beneficial, and we hope there are ways of doing that to bring benefit to patients. And I'll finish there. Thank you.
Okay, thank you. Questions for Adrian? Well,
Great, great stuff. Lipid saturation influences membrane fluidity, and there's a connection to ER stress. I wondered if you see evidence of ER stress in your models and manipulations of saturation modulating that.
Yeah, no, so we haven't looked at ER stress yet. I think saturation does a lot of things. I'd love to talk to you more about that. So some experiments I didn't have time to show did suggest there was an effect on membrane fluidity potentially. And also that's known to have effects of the synapse, of course. So I think it's going to be a slightly painful process now of trying to understand all the different potential effects. But we hadn't thought too much about ER stress, so that's really helpful. That's something we could definitely look at.
Some of the people in this room may remember a movie called Lorenzo's Oil, which was treating adrenal leukodystrophy with not all, because that problem is with longer chain fatty acids in the brain. So I would just cautionary, too much of a good thing may not be good for you.
Yeah, no, definitely. I think there's definitely, we know that the more double bonds you have, the more likely susceptible you'll be to oxidation, and that's going to be a bad thing. So there's definitely going to be a balance, which is why I guess, or I also think the dietary intervention can only take you so far, in that you probably don't want to be overloaded with these polyunsaturated fatty acids either. So I think understanding the mechanism is going to be ultimately the best way to give benefit. But I agree with you. I think there's a balance, and we don't really know now in the sort of rapid progression of ALS whether having those extra PUFAs will be beneficial, and the peroxidation may not be such a problem in the context of that rapid progression or not. But I agree it's something we need to be really cognizant of.
Very nice talk. I have a general question. You show quite nicely toxicity for neurons. But what, for example, if you drive your dipeptide production, not only in neurons, but the general Drosophila driver? Do they still die from neurotoxic syndromes? Or in other words, if you express it, say, in some other cell types, is it also cytotoxic, or the neurons are specifically sensitive to it?
So we haven't done those experiments. I think there is some evidence it can be toxic when you drive in muscle, if I'm remembering correctly. I mean, I think if you drive too much of these really highly charged proteins, they're going to definitely be bad for a cell. So I don't think the specificity is going to come from some cell types being resilient to dipeptides. I think it may more come from where they're expressed. So when we look, in the patient brain, the aggregates are largely confined to neurons. But, maybe it's possible that these other cell types are better at turning them over. So, yeah, that's probably where the cell type specificity may come from.
Thank you.
Yeah, Adrian, great talk. In the second part, where you looked at different effects being observed in the patient brain with the different kind of treatments you had, you made me think of recent work from the lab of Chris Walsh at Harvard, where they have described somatic mosaicism in the brain, particularly in the cortex, and the idea is that perhaps in some cases of the disease can start focally where these mosaic, basically some cells have the repeat and their neighbors don't have the repeat, and then there is disease spreading of DPRs. So I'm wondering, how should we think about that when we want to, for example, deliver Cas13 and target those cells and not their neighbors? What are your thoughts on somatic mosaicism?
So, I'm embedded in a department that has really brilliant work going on in Huntington's disease, including Sarah Tabrizi, who led this amazing trial that shows potential benefit. And there's a lot of interest, of course, in somatic expansion. And I guess my one caution there is that was so clearly driven by the genetics that all of these mismatch repair genes came up. In Huntington's disease, we don't see that signal in C9. And I do wonder whether if you've got 1,000 repeats in the brain, having 1,100 in the brain is going to make that much difference. So at this point, I think the jury's still out on how relevant that mechanism is. And yeah, that's what I'd say.
So, Adrian, yeah, my question was actually highly related to that. Do you know what the germline transmission number is in patients? I remember asking the geneticists that earlier, and they said that it wasn't known.
Well, what do you mean by the
Well, the number of C9 repeats that's actually that you inherited by patients. We know how much is in somatic cells, but just measuring, for example, in male carriers and sperm, you would have. I didn't know that. I don't know if it's ever been measured.
Yeah, I haven't seen any data on that.
Yeah. So my question was, if we accept, and I think we have to, that there is ongoing there's certainly somatic expansion, and probably based on the Huntington work, ongoing somatic expansion. Do we know that what expansion does to the level of these dipeptide repeats, or even the RNAs?
So there isn't, as far as I know, any good evidence correlating length with any of those features, right, at the moment.
Yeah.
So maybe you'd have to do it at single cell level, Don. But in terms of bulk analysis, there's no good evidence that repeat length particularly associates with expression levels or dipeptide repeat levels.
Yeah. Thanks.
But I agree there's definitely, if you look at the cerebellum versus the cortex, you can see a bigger smear on the southern blot in the cortex than you can see in the cerebellum. So there's definitely expansion going on, which I guess is not unexpected given it's so big.
Yeah, so there are some questions on the chat, if you allow me. Nitesh Sangai asks a couple questions. First, he says, awesome work. And I'm curious about the clinical trial failure of ASO despite target engagement from Biogen. Do you think it's because of the duration of target engagement was not sufficient because of the poor bioavailability of the ASO? And as liver acts as a bioreactor for ASOs and cannot cross the blood-brain barrier. So what are your thoughts on that?
So I think there was really nice work just published in Cell from Zach McKeekin and the group in Atlanta, showing that, of course, some did get into the brain. So while there was some good target engagement with soluble dipeptides in CSF, there was less good clearance of the, well, there wasn't really clearance of the aggregates in the brain. So I think there's a lot of potential explanations, right? One is that we just didn't sufficiently get rid of the DPRs. Secondly, of course, we didn't shift TDP, which may well be the executioner in this case. So I think there's just a number of different possibilities. And of course, we didn't touch antisense either. And be very happy to hear Don's view as well.
Yeah, so again, as part of that team that initiated that approach, of course, it was deeply disappointing that it failed. Not only did it fail, although there were measures of target reduction, but there was no clinical benefit at all. And there's so I've been associated with Ionis right from the beginning, and Ionis made the ASOs. Biogen applied them, but there's an intra-team disagreement on why they failed, and Adrian just outlined the various points, and I think it's really confused. To me, and Ionis cringes when you say it, and there was just 2 weeks ago or a week and a half ago, there was the Oligonucleotide Therapy Society meeting. It was in Budapest, and there was lots of enthusiasm for the overall approaches, and now maybe 100 different companies going forward. But I think the evidence is fully consistent that they took forward a toxic drug, and that therefore that on disease mechanism it's closer to silent. Although I certainly agree. Maybe it's really the antisense strand that's the more or antisense strand RNA and/or DPRs. It's just murky. It's really unpleasantly murky.
So speaking of that, Adrian, can you make a comment about any potential advantage of CasRx as in terms of toxicity or delivery relative to the ASOs that have been utilized? It sounds like one potential explanation is that the ASOs were toxic. Do you think that CasRx might be less toxic? There's also emerging evidence that the C9 patients might have heightened inflammatory responses. So,
yeah, I think I'd be happy with either getting to the clinic. Of course, the main benefit of the CasRx approach is the single kind of one and done type of thing. So, but of course, there are also challenges with expressing a bacterial protein in the brain for an extended period of time. So I think both approaches have pros and cons, and I think we just need several shots on goal, basically. So we need to be developing, both of them in parallel.
Yeah. Actually, and I would absolutely amplify that last point. It's a real arrogance to think one strategy is going to be the best, the only strategy. I completely agree that the idea that you were putting forward, it's a good idea and really deserves to be developed. I came back to the podium or to the microphone in part because again, the issues about what's really toxic, it's. I think the cognoscenti really we're confused, and therefore multiple approaches are appropriate. And I really would encourage that, because the simplest ones certainly haven't succeeded yet, and they may not succeed. The idea of targeting antisense strand is actually quite challenging. It makes way too much protein for the tiny little amount of RNA that you can measure. It's really confusing, but you can't deny that the proteins accumulate. That's the evidence from you and from others. It's just overwhelming. So there's lots of things we don't fully understand. Yeah.
Okay.
Or understand at all.
Yeah. This was great discussion. I think we have to move on to a short break. So thank you to both Adrian and Yvette for a great morning session. We'll be back at 10:50.